Thanks, Claudia. Thank you to AdAlta for the opportunity to give a little bit of clinical context, and I'm very appreciative of AdAlta's work in this space. Idiopathic pulmonary fibrosis, or IPF, is a devastating condition. It typically progresses and greatly shortens life, and as such it has a prognosis that is worse than that for lung cancer. And whilst it's designated as an orphan lung disease, it does affect over 300,000 people around the world, with up to 40,000 people dying each year from IPF. And from the point of diagnosis, the patient with this condition can expect a median survival of under 4 years, therefore greatly shortening their life. As reflected in the term idiopathic, we don't truly know what drives it, although epidemiological studies suggest that those who are exposed to cigarette smoke and other dust and environmental pollutants are slightly more likely to develop this condition. But in fact, when you go back to the individual patient, many of them don't report such exposures, and in most patients we can never find a causative link to why they've developed this condition. As I said, it leads to progressive destruction of the lung, and as such patients develop progressive breathlessness, ending up with impaired quality of life, requiring oxygen, and unfortunately typically dying prematurely of their lung condition. And whilst there was great enthusiasm a number of years ago with the development of 2 agents that changed the natural history of this condition, there remains great need to improve on these early phase 1 or MAC1 antifibrotics, given that their efficacy is such that they don't halt the disease, they merely slow the progression of the disease, and it comes with the cost that in over 2/3 of patients they will go on to develop significant side effects that limits tolerability. Whilst a lot of this discussion is primarily about idiopathic pulmonary fibrosis, this is one of a number of fibrotic lung conditions, some of which I'll go on to talk about, but it's worth just highlighting that interest in this area has actually accelerated over the 12 months through the recognition that a number of patients who survive COVID pneumonia end up with a fibrotic and irreversible condition, and therefore there may well be an extended indication for any antifibrotic drug that turns out to be efficacious. This slide just shows some of the activity in the research area with clinical trial activity, and I think it's encouraging that there are a number of biotechs that are in this field, but it's also discouraging in as much as that a number of these groups who may have shown early interest haven't been able to progress through to phase 3 studies and haven't been able to progress through to getting their drugs of interest into the clinics and therefore into our patients. And I think it was a great disappointment that the Galapagos study only recently was prematurely ceased on the basis of lack of efficacy as reported, although the full data hasn't been shown. As you can see from this slide, the importance of FDA recognition is important, and it's, I think in my mind, a key feature that AdAlta have been able to get orphan drug recognition, and that will certainly accelerate their pathway through the drug discovery pathway. So just to summarize that, we're able to show that CXCR4 is upregulated in ILD patients as well as IPF patients. And I think that if it worked in IPF, it is likely to work in the other ILDs as well. And there are at least twice as many non-IPF ILD patients as there are IPF patients. And this also reflects how the field is going elsewhere. Nintedanib, one of the early antifibrotics that did show efficacy in IPF, they've gone on to also show efficacy in some of these other conditions. CXCR4 is regulated in a number of areas, both within the lung itself, in the epithelium, but also within myeloid cells in fibrotic tissue, and that these epithelial cells trigger the fibrosis cascade, and blocking this could have a broader effect than simply shutting down the collagen deposition. So if we can shut down the area where the injury is occurring, all the downstream events may also be switched off as well. And as I said, its effect on some of the immune cells that go from the bloodstream into the lung, and the fact that CXCR4 mediates migration of fibroblasts and other pro-fibrotic cells such as macrophage in a disease-specific way, I think is likely to be beneficial, such that blocking CXCR4 may also have an immune modulation effect and inhibit immune inflammatory cells from infiltrating into the lungs. I think it's also worth just highlighting that this mechanism of action is peculiar to what AdAlta is doing, and as I talked about some of those earlier biotech companies, they were following different mechanisms, so even though they weren't able to get their therapeutic into the patients, that shouldn't dishearten those who have an interest in AdAlta, seeing as we are looking at a completely separate pathway. So I think that, to summarize what I know about the field and also what I've known about the journey that AdAlta have been on, I think that there is no doubt in my mind that there remains an unmet need in idiopathic pulmonary fibrosis, but also in a host of other interstitial lung disease. That moving forward, we're likely to be using combinational therapies on our patients, so it could be add-on therapy as well as solo monotherapy. And I think that the research that we've done at the Alfred suggests that if we target CXCR4 and it works in IPF, it's almost certainly or likely to work in a number of the other interstitial lung diseases. I'm encouraged by the safety data that has been shared with you today, that AD214 is well tolerated and is now ready to move forward into multi-dose studies in healthy volunteers as well as patients, and certainly we'd be happy to be providing our own IPF and ILD patients into those phase 1b studies. And also, again, the patients will be encouraged that the data is supportive of extending that dosing interval to 2 weekly at least, and there's a potential of moving it out further than that as well. So I think their approach is methodical and appropriate, and I think that while we haven't really talked about it, I think one of the key features which is really important is their PET imaging strategy, and there's work out there that if you can see a signal with PET where you have patients who are positive for CXCR4, there is the potential that will identify a cohort of patients who are more likely to respond to drug. And it may also, if you're able to demonstrate that that signal diminishes, you may be able to greatly reduce the length of time that patients need to be studied in phase 3 studies from our current norm of 48 weeks to something a lot shorter, and clearly there's economic benefits in that. So I look forward to the further work that comes from the phase 1 studies. I think that it will be important to see what the CXCR4 receptor engagement in tissue looks like. I think we need to learn a little bit more of the nature of these anti-drug antibodies that, as Claudia says, are expected with a biologic. And I think that will also be interesting to see some of these blood markers to see whether or not that represents a biomarker. And again, if you can identify biomarkers that suggest efficacy, that greatly aids in the development of phase 2, phase 3 studies. I thank AdAlta for their interest and as well as the audience in this area.